Sandra. That will be a hard act to follow, I have to say, but I'll do my best. Can we please have the slides? Thank you. So my lab was interested in genome instability diseases, and that's how we got into microglia. So what I'm going to do today is I'm going to give you an example of one such disease, which is a classical disease in genome instability field, and how it might perhaps affect microglia. So just to start with reminding you that DNA is inherently unstable, and the result of this instability is the formation of thousands, well, actually tens of thousands, up to 100,000 of DNA lesions of completely different chemical nature per cell per day. Now, a high proportion of these lesions could become mutagenic during various physiological processes such as transcription, replication, cell division, and so on, and bring about chromosomal damage, so damage to our genome. And we've developed a number of different DNA damage response pathways that drive the timely and faithful repair of these lesions to prevent genome instability, to slow down cellular aging, and of course to prevent disease. Now, in addition to this endogenous instability of the DNA, there are a number of exogenous factors, usually pleasurable things. I mean, although it doesn't look that pleasurable here, actually. But, you know, when we lie on a beach, then we get UV damage in our skin. And all these factors contribute additionally to the load of DNA damage that has to be repaired. Now, the accumulation of DNA damage is seen in pathological tissues. So, for example, in the brain tissues of individuals with classical neurological disorders and dementias, although, of course, it's not causal in this case, but could perhaps contribute to the progression of the disease. More direct link comes from the fact that mutations in more than 60 genes linked with DNA damage signaling and repair processes actually drives a number of neurodegenerative syndromes. And here, just as an example, as a teaser, I'm showing you a single-strand break repair or base excision repair complex. There's two pathways that converge, hence the complex is the same, consists of 6 proteins, and mutations in 4 of them lead to neurodegenerative and, in one case, also neurodevelopmental syndromes. Amazing, right? So what we're trying to do is we're trying to ask the question, are there any universal mechanisms in all these cases that drive the disease progression? And something happened here. And of course, in the genome instability field, we're still a little bit perhaps behind in that, you know, the major focus and major attention is geared towards looking at what happens to neurons. So now, just going back to the DNA damage response, just to give you a bit more introduction. So it's the complex pathway which needs to detect DNA lesions by various enzymes called sensors, and you will have heard of PARP1 inhibition being used for driving synthetic lethality of homologous recombination deficient cancers. So this is PARP1 is a sensor of DNA damage. Then the information is conveyed to transducers, and these are usually protein kinases, such as ATM, which then drive phosphorylation of a number of different proteins, including, again, a range of different kinases, p53, tumor suppressor, and thus drive repair, cell fate decisions, and all other processes around it, which are intended to repair DNA damage in time and correctly. Now, the protein that I'm going to talk about today is a transducer. So it is a serine threonine protein kinase called ATM. This kinase is mutated in a disease called ataxia telangiectasia. I had to practice this one for a few years before I got it right, but now I think it's perfect. I'm still going to call it AT. Now, this kinase is important classically for DNA damage response to DNA double-strand breaks, which are cytotoxic because we can lose information at the ends of these double-strand breaks, but it seems to also be involved to repair of oxidative damage, single-strand breaks, and so on and so forth. And hence it plays many different roles, perhaps not directly, but through its substrates and regulation of those. Now, going to the disease, as I already mentioned, AT is caused by loss of function mutations in ATM, and it has a few different phenotypes across the body, but in the brain, it manifests as neurodegeneration, specifically actually in the cerebellum. So here you can see cerebellum of a healthy individual, and here is the cerebellum of an individual with AT. And this is a 7-year-old child, just to give you a comparison. So these are age-matched pair. And you can see huge loss of cerebellar matter, which results in the phenotype, which is these kids are they lose the ability to motor control and to coordinate their movements, and they're actually wheelchair-bound by their early teenage years. Now, the fascinating thing about ATM loss from a molecular perspective is that, and here I'm giving you an example from the Wasserman lab, and this is a Drosophila model of AT, whereby they knock down ATM in the whole brain and observe typical neurodegenerative phenotype, well, as typical as you can get in a fly. And if they knock down ATM in neurons alone, then there is actually no neurodegenerative phenotype. And this is the first sign that non-neuronal cell types play a role in disease development. So now, if we look at the level of expression of ATM in different cell types in the brain, then it is actually the highest in microglia. So the bigger the dot, then more efficiently we detect ATM, and the redder it is, the higher the expression levels are. So we decided to focus on microglia and ask a very simple question. What happens to microglia cell intrinsically if we remove ATM from the system? Are they different? And would they obviously affect neurons in any way? And what we find, I'm going to disclose my secret straight away, is that, again, I'm using a simplistic kind of approach to describing what microglial states here, but normal or homeostatic microglia, upon loss of ATM kinase, change. And this change results in effectively loss of regulation and damage of neurons. So just to give you a little bit of more details, data, hopefully you can see it. So these are ATM knockout neurons. They look lovely. If you can see here, we're labeling with beta-3 tubulin, we're labeling neurites, and DNA, so stained for DAPI in blue. They fire well, so they're absolutely fine. Now, if we add microglia to them, I hope the sound is not too loud because I think I have some sound here. So what happens is the neuronal network doesn't look great. And here I'm giving you an incredibly, perhaps exaggerated example of how bad it is. And in this particular example, we see a cluster of microglial cells and a lot of damage around this cluster, loss of neurons, and also damage to neuronal structures. And if we zoom, and effectively that's what it looks like. Pac-Man's been here. Oh, good, there is no sound effect because there was music as well, the classical music that we all love. So now if we zoom into this cluster, you can see that it is incredibly phagocytic, and there are lots of phagolysosomes containing all sorts of material, including pieces of, again, neurites, I would describe them, so beta-3 tubulin positive material, which is DAPI negative, and double positive. And we can actually quantify overall damage to the network, which is existing, right, but not huge. We can also quantify this, what we call phagocytic clusters that are associated with great damage to neuronal network around them. And we see that these clusters do not form in microglia-only cultures, but there is a huge formation of this damage around microglial clusters in knockout cultures versus wild types. So what we decided to do is we said to ourselves that why don't we, dissect, again, in a very simplistic way, this microglial dysfunction and this damage that seems to be coming from microglia into a few different phenotypes and try to work out how they're regulated. And we've dissected it. That was over breakfast, so maybe it wasn't, looking back, but simple and direct, right? So we dissected it into aberrant phagocytosis, because what we actually see here, fascinatingly, is that ATM-deficient microglia don't really discriminate the type of substrate that we give them. We can give them ATM knockout neurons, we can give them wild-type neurons. They just eat and eat and eat. And this is true also in tissues with individuals with AT, where a lot of phagocytic genes are greatly upregulated in the cerebellum. We're not sure we know how this is regulated, but it's fascinating. So we call it aberrant phagocytosis. Then clearly, clusters clearly provide greater damage than just, microglia alone. And to form a cluster, they need to get there. So why don't we look at chemotaxis in this instance? And also, of course, we have overproduction of a number of pro-inflammatory cytokines and other molecules that could potentially be neurotoxic to neurons. And in terms of how it would be regulated, well, it would be low-grade inflammation, we thought. It would be upregulation of some inflammatory pathways. And we were looking for those that, cytokine profile gave us some clues. We were looking for things that could be active in the brain and also that could potentially be regulated by ATM absence or perhaps by genotoxic stress associated with ATM kinase loss. So one pathway immediately comes to mind is an F-kappa B. So interestingly, it's not a classical canonical F-kappa B pathway that is activated in ATM-deficient cells, but so-called non-canonical, or better defined perhaps as REL-B p52 heterodimer-driven pathway. So an F-kappa B pathways in general drive inflammation very similar to SIGA STING driving type 1 interfering response, which is antimicrobial. And F-kappa B pretty much does the same. Does it also downstream of STING, of course? That was as was described by Andrea. So they drive inflammatory responses, cell survival responses, and this particular subpathway also seems to have some roles in specific cell types, which again, cell type specific, such as differentiation, for example, of dendritic cells, maturation, the other way around, differentiation of T cells, maturation of dendritic cells. We're not sure what it does in microglia, but again, we're working on it. Now, how does the pathway work? In general, an F-kappa B is not active. It is sequestered in the cytoplasm in this inhibitory interaction with a precursor of p52 protein p100. And there's also a persistent degradation of E3 proteasome-dependent persistent degradation of the NEAK kinase, which is an upstream mediator of this pathway. Now, usually upon receptor stimulation, the interaction between the E3 ubiquitin ligase and NEAK is disrupted. NEAK phosphorylates another kinase, that phosphorylates p100, and that drives proteasome-dependent cleavage of p100 into p52. That exposes the nuclear localization signal on the REL-B, and the heterodimer goes into the nucleus and drives cytokine response. So it's a very nicely controlled system, and there are ways to turn it off as well in response to this acute inflammation driven by various stimuli. So just to give you, again, a very quick and simple snapshot of what happens in ATM knockout microglia, we see relocalization of REL-B in ATM knockout microglia in basal conditions in the nucleus, and that happens in about 50% of the cells versus about 7.5% in wild-type conditions. We also see accumulation of REL-B and p52 in the nuclear extracts of ATM-deficient microglia. Now, this is in vitro. Does this happen in real individuals with AT? Well, actually, it does. So here we teamed up with Alice Lee and Mike Lodato, who had a single-cell RNA sequencing data set from individuals with AT and matched controls. And we've asked them to basically look at the non-canonical and canonical F-kappa B pathways specifically in cerebellum microglia. So what we're looking here is an average T-score, which is effectively a measure of change in expression. change in gene expression in different conditions. And this bar, so if we took, for example, a data set of the same size as this non-canonical and F-kappa B data set, then that data set would give us a distribution of t-scores shown here as this bar. Now, the actual t-score of our specific data set or gene pathway, gene set, is shown here in red. And what it means is that this particular gene set is significantly upregulated in AT microglia versus healthy microglia in the cerebellum. And this is true for positive regulators of non-canonical and F-kappa B, such as, something interesting is happening here, such as REL-B, B100 gene, NIC, and some receptors as well. And we don't see really any significance with negative regulation of the pathway. What we also see is that there are no expression changes that indicate activation of the canonical subpathway. So now this is all lovely. Again, tells us that it's true perhaps in vivo. Well, not perhaps, but it is. And what we actually find is that this pathway, first of all, drives the production of a number of classical and F-kappa B cytokines, some of which could be neurotoxic. TNF-alpha is the one mentioned by Andrea, but also IL-6 and IL-8. In addition, it actually drives the increase in this aberrant phagocytosis. And what we can do, this increase in phagocytosis can be rescued back to pretty much wild-type levels by knocking down the REL-B subunit of this heterodimer, which is true for ATM knockout microglia, but not for wild-type microglia. So what I showed you very, very briefly is that ATM-deficient microglia show this systemic inflammation phenotype, which is driven by the REL-B, P52, and F-kappa B pathway. Well, it would be lovely if it was the only one. Of course, it is not. And this is where Andrea very nicely introduced the SIGA-STING interferon response. And of course, the SIGA-STING interferon response is also activated in ATM deficiency because ATM deficiency is a genome instability, effectively phenotype. So the pathway has been very nicely introduced, but what I wanted to just remind you again is what could be the sources of cytosolic DNA formation in general in genome instability and specifically in ATM deficiency. So Andrea mentioned already micronuclei. Micronuclei are formed during cell division in the presence of DNA damage. In addition, of course, during repair or inefficient repair of DNA damage, we would always have pieces of double-stranded and also single-stranded DNA, which could leak from the nucleus when a nuclear envelope breaks down during mitosis. In addition, it is proposed that there is some active export of such species into the cytoplasm, supposedly for degradation by, for example, TREX1 and DNAs that were mentioned again by Andrea previously via exportin 1, although that could be, again, because exportin 1 is highly promiscuous and it binds, actually exports lots of DNA-bound enzymes and proteins. Now, mitochondrial stress, something that's been mentioned previously, and for example, R-loops, which are effectively physiological intermediates of transcription, but could, again, be released into the cytoplasm. And while we cannot exclude that, many of these cytosolic DNA sources actually activate the SIGA-STING signaling pathway in ATM deficiency, one thing we can actually detect very clearly is SIGAs at micronuclei and the downstream interferon signaling. So if you look here, then we're looking at a couple of examples of nuclei with micronuclei next to them. And this micronucleus is SIGAS negative. And here you're looking at a micronucleus that is SIGAS positive. Now, the number of micronuclei increases in the absence of ATM, and this would be true for many genome instability disorders, so long as we have proliferating cells. And within this micronuclei subset, some of them are cGAS positive. Now, this drive the downstream type 1 interferon response, and again, I'm just giving you a couple of examples that the pathway is activated. So what we're doing here, we're using actually a brain-penetrant ATM inhibitor, which is actually developed by AstraZeneca, to look at the temporal activation of the pathway downstream ATM loss. And you can see that it doesn't happen instantly, it takes time. Yeah? So after 6 days of treatment, we're actually beginning to see by Western blot measurable activation, and it's probably amplified a bit more by more sensitive methods of detection. And what you can see here is this phosphorylation of STING, which indicates its activation. You see phosphorylation of TBK1 kinase, which is upstream of STING, in the same complex, and also phosphorylation of IRF3 transcription factor, which then results in its relocalization into the nucleus and type 1 interferon response. Now, downstream of this activation is type 1 interferons being produced, interferon-stimulated genes, cytokines, chemokines, the whole bunch of them. Now, we can block this by blocking mitosis with CDK1 inhibitors, so we don't allow cells to divide. And we can prevent this response, which indicates that perhaps replication, DNA replication is important, endomitosis, so it's hard to discern here. We can also block this response by knocking down cGAS, which means that it is this canonical cGAS STING dependent type 1 interferon response. So now going back to our microglial phenotype after all this diversion, I think we had chemotaxis left, right? So you can see where I'm going. I'm just going to tell you now that cGAS STING controls chemotaxis, and indeed it does. So this is a collaboration with the AstraZeneca team led by Mike Perkington, who's here in the audience. And what they've done is they've taken microglial cells, treated them with interferon beta, so receptor-driven activation of type 1 interferon response, and analyze cells by bulk RNA sequencing at a couple of different time points. Now, if you look at enrichment analysis of different pathways, then what you see shown here in different color is lots of different pathways associated with cell motility, cytoskeleton rearrangements, and so on and so forth. So what we did is we then looked, with help of Calliope, looked at individual genes that are in these gene sets, and we find actually a huge cluster of chemokines that is forming in time and very, very apparent and separated at 24-hour time points. And we decided to look a little bit further into one of these genes, which is really, really bright red. I'm sorry, there is a bit of a delay on my mouse here, which I don't see actually on my computer. So it's CXCL10 or IP10. And what we can see is that ATM loss stimulates the production of CXCL10. We can stimulate the production of CXCL10 to a certain extent by treating cells with STING agonist, and then we can sensitize ATM-deficient cells to produce even more by this additional STING agonist treatment. In STING knockout microglia, this chemokine is not produced, and this is true for a great range of different chemokines, CCL and CXCL proteins that we looked at. Now, does it translate in actual migration changes? It does. So here we're looking at chemotaxis migration assay. We're taking either wild type or STING knockout microglia in the presence or absence of ATM inhibitor and allow them to migrate towards their respective conditioned medium. And what we see is that ATM deficient cells, let me find them, here we go, here, compared to so this orange curve compared to the gray curve, chemotax quite a lot more, and this is partially prevented by knockout of STING. So I'm pretty much finishing up here, just to say that we seem to have ticked all our questions, in this little project. And what we're saying is that ATM-deficient microglia seems to drive this low-grade inflammation via two different pathways, which regulate, co-regulate each other, but also seem to act independently of each other. So I think there's a great complexity actually there. And that can also drive microglial dysfunction. Now, the result of course, production of huge numbers of different pro-inflammatory molecules is that we're going to have huge numbers of positive feedback loops through a number of different receptors, which I'm mentioning here, and set off this inflammation and propagate it. Now, one thing I wanted to quickly mention is that we can drive this change by just treating cells with very, very low levels of a variety of different DNA damaging agents. So we don't think it is specific to ATM loss. We think it's a feature of genome instability, as fairly clear from, for example, microglia formation. And here again, as an example, we can see REL-B nuclear relocalization upon treatment with etoposide, or we can see sting-dependent accumulation of another chemokine, CCL5, in response to etoposide treatment. So yes, so basically what we think is that persistent DNA damage drives low-grade inflammation in microglia, and that adds to the complexity of disease development. Of course, ATM DNA repair deficient neurons will also have problems, so there will be an interplay between neuronal states, microglial states, and all other things in the mixture. Thank you very much to my lab. So Julie is my first postdoc. She's now an associate professor back in France, very happy for her. And she's done the work on REL-BP52. And Emily is my first PhD student who's just graduated, and CGASting is mainly her work. Thank you very much.
So do we have questions?
Thank you for that. That's really interesting to me because it's incredibly similar to a phenotype that I'm seeing with some knockout microglia. So I'm interested in a protein called oxidation resistance 1. And in patients with loss of function mutations and in mice, we see specific degeneration of cerebellar granule cells and sort of cerebellar atrophy. We're not sure whether this is because it's at a developmental time point, so this occurs in sort of adolescence, and in sort of P20 in mice. We're not sure whether it's, there's definitely a neuroinflammatory aspect, so we see activated microglia around the neurons, but we don't know whether it's a neurodegenerative or a sort of perturbed development of the cerebellum, because it's so early on, right? So.
I mean, it's a great comment. I'm not sure I can actually answer this question because we don't, I mean, again, you know, classically we think that many of these diseases are actually neurodegenerative, and that's how we define them. But it's very hard to imagine that, you know, any type of DNA damage in neuronal progenitors, or in fact in microglia, for example, erythroamyloid progenitors, would not bring about some early problems. There's certainly something there, but again, we don't have any good evidence of, you know, what DNA damage or oxidative stress would do to progenitors. I think we know a bit more about neuronal progenitors because, I mean, they die in p53-dependent manner like mad, but some of them we know accumulate some somatic mutations that are carried through, and some of these actually do result in later problems. There's certainly a neurodegenerative component because, you know, if we take, for example, a mouse model of DNA damage, they usually don't have any neurodegenerative phenotypes. If we now cross this model with another DNA repair deficient mouse, then we would amplify the phenotype. So it's this load, ever increasing load of DNA damage that seems to be driving the phenotypes.
And one more quick one. How did you manage to get siRNA into the microglia? Because when we've tried to do it, they just gobble it up.
Not easy, but we're using, I think it's a Fugene, which I think is a magic, absolutely magic liposomal delivery drug. I have no idea what's inside. Okay. And yeah, that worked.
Okay, cool.
And, you know, we try to avoid virus delivery. as much as we can.
In many cell types, cGAS has been identified as a regulator of senescence. And I was wondering whether you had the chance to test for senescence markers and if the inflammatory profile you have identified is more like a senescence-associated secretory profile as compared to a real output of a high volume, high concentration inflammatory signal.
I mean, it absolutely is. The problem with SUSP is that it's very similar to DNA damage response. So it's hard to distinguish between them. But yes, everything that we see could be attributed to senescence. Now, we've tried to look at senescence in our core cultures and things like this. What we'd really like to do is to be able to identify it in actual tissues of individuals with AT, and I have no doubt that there are senescent phenotypes there.
Short question, short answer. Here.
Thanks for the talk. I was just wondering if you considered looking into other immune cell types about this pathway, given that patients with ataxic telangiectasia seem to have immunodeficiencies, and it sort of seems that there might be a difference between how this works in adaptive versus innate immunity, or is one driving the other kind of thing?
I mean, absolutely great question. So yes and yes. And one thing we're actually looking at is we're looking at whether some of these responses would drive the infiltration of actually peripheral immune cells into the brain and then amplify the phenotypes and/or
Good. Thank you again.